Hi, kiddos. So these are the last three learning targets on the Learning Target Tracker, Unit 7, Number 5. And these all have to do with the BLAST lab. So I just wanted to make sure that you knew how to use the concepts from the BLAST lab and how to kind of interpret all that. So the three I can statements are, let me read them. I can create cladograms that depict evolutionary relationships. We're actually going to do that last. I can analyze biological data with an online bioinformatics program. That is BLAST. So basically, I can analyze biological data with BLAST. And I can use cladograms and bioinformatics to ask other questions and test my ability to apply these concepts to answer those questions using genetics and evolution. All right, and that last one was really what you did with your protein. So you were given a protein, you had to get the sequence for it, blast it, and all that kind of stuff, and then you analyzed an evolutionary relationship based on the genetic similarity. So when it says, I can use cladograms and bioinformatics to ask other questions and test my ability to apply these concepts to answer those questions using genetics and evolution. The genetics is that percent similarity. I think it's called the query cover. And then the evolution is the cladogram. So basically, I can use the cladograms and the information I get from the BLAST site to apply that to a new situation with your protein that you were given, and then use what I learned from that protein, whether it was ubiquitin or polymerase or whatever it was. Then you can use that information to deduce how closely related organisms are. And so you were able to look at the query covers and create a new cladogram. So that's what that last one is talking about, is basically you taking the information you learned from the beginning of the BLAST lab and applying it to your protein. So I'm not going to really sit on that one much, but I am going to run you through that process. So what I've done is I've gotten a DNA sequence, magic P-row there, got a DNA sequence. I'm going to blast it, and that's going to take a minute so we can sing the little Jeopardy song to yourself if you want. And what we're going to do first is actually the second one I mentioned. We're going to analyze biological data with an online bioinformatics program, or BLAST. So what we're going to do is we're going to look down here, and actually let me open this guy. And whoopsies. Let me see if I can shrink it down a little bit, because I really want to be able to see as much of this as I can. Okay. So what I've got going on here is, I guess this sequence was for the angiotensin converting enzyme. So what I see is it's 100% query cover. If I look at the distance tree of results, this is a cladogram that it makes. Don't want to give my feedback. You'll notice that all of the dots and the little, I don't know, triangle looking things are green. And if you look over here, green is primates. And so what we know is that this gene is found primarily in primates, or it has the most similarity with primates, which makes sense because it is human and we are a primate. So, sorry, I had a little interruption. And so that's one of the ways that you can analyze your data, is you can look and see where does BLAST put it on the cladogenetic tree. That's not a thing. The phylogenetic tree of the cladogram. Anyway, another thing you can do is you can look through and you can see who else is closely related. And this is one of the things you did with your protein. So I'm going Homo sapiens, that's us, so I'm not going to write that one down. The next one is this troglodytes. Don't know how to say that. I don't speak whatever. Latin. Panpaniscus. Oh, I need to be writing down the query cover. Query cover is 100%. Panpaniscus is 100%. And then you can also look at the percent identical. So that's 99 if you need, like if these are both 100, and 99.37. So there is a little bit more identicality, I don't know what word, with this one. And then the next one is gorilla, which is shockingly the gorilla. And that's at also 100% and at 99.22. And then we'll just do one more. I know in your lab you had to do 5, but we're going to be lazy and only do 4. And that is at 100% and 98.32. Now, if I wanted to take the time, I could look up that. So that pan troglodytes, whatever, is the chimpanzee. And, whoops, pan paniscus is the bonobo. Or you might hear it called bonobo. Gorilla is the gorilla. We'll go ahead, since there's only one more, we'll go ahead and look that up. Pongo, yeah, is an orangutan. So this is the gorilla, and this is the Sumatran orangutan. Orangutan, actually. All right, and so what I would do is I would use this genetic information to create a cladogram. And so coming off the top is going to be my humans, because we are the most recent to diverge. And then I'm just going to put them in the order that they are, from the most similar to the least similar. And so my chimpanzee would be the next one, the closest to the human, because it has the number that's the highest, has the most similarities. And then my bonobo, then my gorilla, and then my orangutan. And that's how you would make a cladogram. If you wanted to make a phylogenetic tree, you would take the one that comes off first, or has the least similarities. That's the orangutan. And then you'd have the gorilla. And then you would have the bonobo. And then kind of coming off, whoa, that's an ugly line. Coming off together there, you would have the chimp and the human. So if they wanted a phylogenetic, I just realized y'all didn't see any of that because we were looking at the orangutan. Sorry. We'll look at it here. So if you, orangutan's a lot cuter than a cladogram anyway. If you did it by a cladogram, it would look like this. So you have your humans at the top, because they're the most complex, the most recently diverged. And then you just go in the order from most similar to least similar. Or if you wanted to start here, you would go least similar to most. And then with the phylogenetic tree, you branch off the least similar first, and then the next one, then the next one, and then since chimps and humans have the most similarity, they would branch off together. Okay, so that is how you would create a cladogram that depicts evolutionary relationships or phylogenetic tree. Remember, in AP Bio, we use those terms interchangeably. And it's also how you analyze this data, this genetic data about these organisms, and use it to determine evolutionary relationships. And that's it. So if you have any other questions, please let me know. Thanks, guys.